Welcome, everybody. It's exciting. It's our first time back in person for an IFAM seminar in quite a while. Forget the count now, but it's been over a couple of years. So excited to have at least some of you back in person. And we are doing a hybrid format today, so hopefully that will go well, meaning that we have people attending by Zoom as well as here in person. I'm Ron Ackerman. I direct the Institute for Public Health and Medicine. I want to welcome you today to our first IFAM seminar here in person. And as just logistically, not for you all in the room who can continue to enjoy your lunch, but for those on the phone, we will continue to operate the same webinar format where if you do have questions, we encourage you to submit them, but you must use the Q&A function on the bottom of the Zoom screen. If you're in person, you can just raise your hand, which is really great. If you're online, please don't use the chat function because we won't be monitoring that. And we will open the questions and introduce them to our presenter today at the end of her talk. It's with great pleasure that I am able to introduce Dr. Leah Welty. Dr. Welty is professor of preventive medicine and biostatistics and psychiatry and behavioral sciences here at Northwestern University's Feinberg School of Medicine. She's director for the Biostatistics Collaboration Center and also is considered an expert in the application of biostatistics to psychiatry and environmental research and the development of software tools for reproducible research. Today, Dr. Welty's talk is part of a recurring series that we offer jointly with the New Cats Institute here on campus called Translational Applications in Public Health. This is the first presentation in the series for the year. And Dr. Welty will share with us her work on the topic of the epidemiology of preventable analytic errors, a scoping review of retracted articles in clinical and translational science. With that, I turn it over to you, Dr. Welty.
Thank you, Dr. Ackerman, for that really nice introduction. I can't tell you how awesome it is to be talking to a room full of people, as well as people online, instead of just people online. So I'm really, really excited and delighted to be here today and share with you some of this work that's happened over the past couple of years. I do want to acknowledge my colleagues before I get started. The work I'm talking about is largely to their credit, both in terms of some of the ideas we had, as well as the sheer volume of review that needed to be done. So I want to make sure to acknowledge them all. They are Abby Baldridge, Grace Bellinger, Oriana Fleming, Luke Rasmussen, and Eric Whitley have all been involved in this. And it's been largely funded through the New Cats Institute. Okay, so this is kind of like the ideal world, how we would like research to go. We have a question, we get some data, we do some analysis, we write a manuscript, and we disseminate it. And ideally, that process takes months, not years, right? And the birds are chirping and the rainbow's out and everybody's happy. And that's not how it works in real life, right? This is more what really happens, right? You have data, and if I had a font that was messy, I would put it here. I just don't know what it means to have a messy font, because data is often messy, and sometimes it's getting messier. So we have messy data. Sometimes we have lots of it. We do analyses. We're working on a manuscript. But maybe as part of that process, right, we're going back and we're saying, look, I think there's an error in the data, or look, I think there's an error in our coding, or look, there's something else we need to think about as part of our analysis. So there's this feedback loop that goes back through it. And that process often takes years. It's also the case, too, that we may, during that process, have people moving in and out of our research teams, right? We have people come and go, students graduate, they become really, really useful to our projects, and they leave us and go on to great things in the world. So, you know, there's these transitions too, and it all is a lot to keep track of, especially now as data gets bigger and more complicated. So Luke and Abby and I were working and are working on something called StatWrap, which I'm not going to digress into here, but I do want to sort of tell you a little bit about it to set the stage of what we decided to do. And this was software we're developing to really manage some of that variation and change in the research process. so that it's a little easier to tell all the decisions that were made along the way and who contributed to them. So for instance, one of the things StatRep does is it provides you this nice directed graph. On the right-hand side, you'll see of how your code files and code libraries are attached to each other. So that you could look at this and be like, oh, I've got this R file and it's pulling data from here, but it's also pulling data from here. And you could check to make sure, for instance, are those the right data files? Then on the left, you'll see something that we call our project log. And this is really just a place to store some of those little random decisions that happen in the research process, but that may not have a home for the meantime. So, you can say, oh, I added this person, or, oh, we made this decision, or, oh, I updated the data and it's here. Whatever you need to log, it's there. It's kind of like just notes. And this is called StatRep, and it's actually pretty far along in development. So if anyone would like to be an early adopter, please email me or Luke, and we would be delighted to have your participation. I know the Zoom bar is covering it a little bit there, but you can Google Leah Welty at Northwestern and you'll find me. Okay, so in this software design process, we were really thinking hard about what kind of errors are we trying to prevent? And to do that, we thought about what kind of errors can happen. So if you think about sort of chopping things up along the spectrum where we have data involved, when you're collecting data and storing it, sometimes people can't find their original raw data, or maybe someone uses the incorrect version of data. If we're thinking about data cleaning and preparation, there's a pretty classic error that I tend to refer to as a 0-1 miscode, where maybe somebody codes yes as 1 and no as 0, but instead it was reversed and nobody knows until kind of far down the line, or maybe never. Or maybe there's sort of incorrect exclusion and inclusion criteria applied to the data set for analysis. Now there can be errors in the choice of statistical methods, right? And I get really wound up about these. I want to make sure that we're using, for instance, methods that account for paired or correlative data, if that's the type of data we have. Then you can also make an error in the application of the statistic method. So not just the choice, but how you've actually put it into use. So maybe there's some incorrect variables in the model, or maybe there's a programming error in sort of how some of it is executed. And then of course you can have an incorrect selection of analyses coming from those analyses. Maybe you've done them correctly, but you generate a figure, you tweak it a little, you generate a new figure, you update your data, generated a new figure, and maybe you don't get the right figure in that manuscript. So these are some of the things we could think of in Brainstorm that we're trying to prevent. But as we all know, this is just speculation. It would really be helpful instead if we had data and we really understood what was going on. So we went down the rabbit hole in a pretty serious way. And we thought, well, we know there's some data out there. And in fact, there are collections of retracted articles. And for anyone who's ever been through that process or seen it happen as an editor or a journal reviewer, it can vary in a lot of ways. But what generally happens, to make sure people are familiar, is somehow there's some type of error discovered in a manuscript that's been published. And there's a lot of places, a lot of people who can discover it, and a lot of ways it can be discovered. In this case, this is an example of retraction that occurred in the American Journal of Obstetrics and Gynecology. And there was a letter written into the journal where some other readers said, we think this article is wrong because we redid the analysis on the data that was publicly available, and there seems to have been a mistake. And the authors who wrote the article then published a reply and said, yep, you're right. In our logistic regression analysis, we mistakenly used the incorrect code for skin incision type. And so the article was retracted. And this was actually a case of kind of a good retraction if you think about it. It was well documented. People knew what the problem was. They discovered the problem. The authors then said, oh, you're right, it is a problem. And the scientific literature then self-corrected. The journal was involved, they retracted the article, they published a notice about retracting the article, and so hopefully this didn't make it into clinical practice for too long. Now there's actually something out there called the Retraction Watch database. Has anybody heard of this? Few hands. Okay, so there's actually a database of retracted articles out there. And it was started a number of years ago, but it's now partnered with the Center for Open Science. And they actually have places where you can look up and see what articles are retracted, and you can get lots of data on retracted articles. So we contacted them and were able to get a full data set from them with a data use agreement. And one of the cool things they do is they give authors title, the reasons for retraction, the subject areas, the countries, and some other things they categorize. So it's actually a really, really extensive database. It goes far beyond medicine and biology. You know, there's engineering, there's physics, there's chemistry. There's basically any place anybody publishes journal articles. And they also give some reasons for retraction. So when those articles are retracted, there's just a notice the journal puts out and said, we're retracting this article because the data was wrong, or we're retracting this article because people didn't get IRB approval, or whatever it is. And so Retraction Watch categorizes these. And there's been a lot of focus on these where misconduct has been involved, falsification, misconduct, malfeasance, sort of unethical behavior. But what we did is said, you know, we're not terrifically interested in those. Certainly they're a problem, but we're interested in kind of more of these honest analytic mistakes, where you think you've got the data right where you don't, or you think you've got zeros correctly coded, but you don't. So we used the database. We created a scoping review protocol that we've actually published online at Digital Hub, Galter Library, big fans of Digital Hub, that follows the guidelines on how to conduct a scoping review. And for this, I want to credit, oh, got cut off there, Q Eileen Wofford of Galter Health Sciences Library for helping us with that protocol review document. And so this is where we landed. So we extracted from the Retraction Watch database all research articles published through March 12th of 2020. So just shortly before we all went into hibernation. And then we sort of scoped them down. So we said, well, we're going to include anything we think is related to medicine, anatomy, physiology, biology, human health, public health. And we omitted things that we were pretty sure weren't the types of articles we were interested in, sort of the space we work in. So agriculture, veterinary science, for instance. Then we included reasons for retraction that were related to data analysis and results, but we excluded things that were like falsification, malfeasance, paper mills, things like that. Then we subsetted to published in 2010 or later. And then came some of the really hard part, because we said, you know, what we really want to do is conduct a review of anything in clinical and translational science. And so that's really because we wanted to say, what are the articles that might be impacting human health that we all have an interest in? And so we actually did a big abstract review of those roughly 1,200 articles to get it down to 884 of them that were in English and related to clinical and translational research. People have done sort of some of these dives into articles before, although not particularly for these reasons, but in specific subject areas, cardiology, cancer, cell biology. But we really wanted to be pretty broad here. So we were saying, look, if it has to do with human health, we're going to grab it. And then we went and said, after the abstract review, we did a full text review of both the retracted article and the retraction notice. And that was on 786 articles. So this is just to give you a sense of what the scientific area looks like for those 786 articles. This is per the Retraction Watch categorization. So you can see there's a lot up there with cell biology. Goes all the way down to sociology and psychology. I will note that I am not in any way trying to implicate people in those fields. This may also in many ways just represent the quantity of literature in those fields too, right? If you have a lot of literature, you may have more literature retracted, and so more journals may fall into those categories. But just to get a sense, everybody can look up there and say, you know, is my favorite area of inquiry up there. Now, here are some of the characteristics that came from our full text reviews. Median 6 authors, range was 1 to 37. 38% of articles had an author attribution statement, which we thought was pretty interesting. Not many people have looked at this before, but you know, to say who's responsible for your data, who's responsible for your data analysis, who's responsible for the interpretation. So that's a number I'd be pretty interested in comparing to the unretracted set of articles out there. I just don't have that number. And then you can see we've got time from publication to retraction here. Generally, it's happening pretty soon, but we've also got some censoring in our data set, right? Because, you know, if you publish something in 2019 and we pulled the data in 2020, if it's been retracted now, we wouldn't know. All right, so here's some of the characteristics we were particularly interested in. Who was collecting the data? Was this, or are these articles using existing data or not? And for the vast majority of articles, that is de novo data collection. 12% was previously collected for research purposes. So they were using existing databases, you know, sort of data sets that had been previously published elsewhere. And 11% was previously collected not for research. So for instance, that might be something like data from an electronic data warehouse that was, pardon me, medical data not. Are you hearing me on the microphone still? Were you hearing me here? Okay, sorry. All right, I thought I was on a different microphone. Okay, so previously collected not for research purposes, maybe some other type of registry, government data, something like that, but it was never intended for research necessarily. Is the data available? 89% of those articles, it was unclear or unknown whether or not people had made the data available in any form. 8% publicly available, 3% by request from the authors. So pretty startling here, I think, especially in light of some of the policies you may have heard about the NIH requiring data sharing for grants that go in after January of this year. So pretty interesting numbers. And then how was the data captured? And this isn't something we necessarily ask people to put in articles now, but we were pretty astonished that there wasn't more mention of sort of where the data was being stored, how it was being housed. About 3% of articles said they were putting their data in something like an Excel spreadsheet or a comma separated value form or a Google sheet. And about 2% said they were using REDCap, some sort of clinical trial management system or a database system. Now, my hope is that there are a lot more articles that were actually using things like REDCap to house data, or CTMS or SQL or Microsoft Access, but people just weren't reporting it in articles because it's not something that we traditionally report. All right, and here's some information about study design. So about a quarter of the articles used live animals, and about 3/4 were human subject space. I will note that we allowed articles to be categorized as both. Some of them did include animal data as well as human subjects data. For those human subject studies, about half were observational, a third were benchtop, where they were cells, tissue-based, and about 14% clinical trials. And then we also had a few meta-analyses in there that were separate. Yes, question.
You say full text, you mean like humans read them, looked at all this?
Yeah, that's a great question. Thank you. The question for those online is, when I say full text review, do I mean actual humans read the articles and input this information? I do. And those humans are the humans that are listed on that first slide. We had to grow our team at some point to recruit some graduate students to help us do that because it was a lot of full text review. So 5 of us reviewed 786 full text articles to extract some of this information. So yeah, if we could have thought of an NLP way to do it, that would have been great, but no, we did this by hand. So I want to play a little game with most common statistical software. I want you to think about the statistical software you use in your practice, in your lab, in your analysis. And so think about what you think these might look like. I'm going to present them in the order from some of the less common ones to the most common ones. I'm not going to give you the really fringy ones that were like less than 1%, but I'm going to start at about a 1.5% or higher. So get your favorite software in your head, and maybe even think of the one where you're like, well, that's bad if people use that. It differs by field. I'm not judging. All right, 1.5% of these articles purported to use Excel, Google Sheets, or some sort of spreadsheet program to do their statistical analysis. For those of you who've seen any of my other talks, you know that I recommend against this strongly for a number of reasons. Excel was never designed to do complicated statistical analysis in any way. It has this funny habit of cutting off data if you have too much of it. It also does this thing where it takes the names of genes and it thinks they're dates. So Excel, not so recommended. It's also not really reproducible. So think of your next favorite one, maybe one you've heard really common and you think like, wow, people use that, they really know what they're doing. Yes, thank you for the gasp in the room. For those of you online, there was a gasp in the room, and this was the same reaction we had. We're like, oh my gosh, this is terrible. So as many of you know, R and R Markdown or R Studio it's a very well-known and popular statistical package. It's known for reproducibility. It's very extensible. It's open source. There's a lot of use of it. You know, I think it's taught in many of our statistics classes here at the university. Yeah, 3.7% of articles. So maybe we'll go on to some other common statistical software. SAS, less than 5%. Stata, about 5%. Okay, so now I'm getting a little bit worried because I consider R, SAS, and Stata like my three main statistical go-to programs. Like they're the ones I believe and I trust and I'm really, you know, confident in my ability to work in. And I'm like, wow, what are people doing? Well, there's GraphPad Prism, which I think is really, really common in some basic sciences, especially. I'm just not as familiar with it in the statistical world. And then there's SPSS products that are near 30%, which was really surprising to me. One of the things that always concerns me about SPSS is, although it has a lot of statistical sophistication, it's a very point and clicky type of program, which means that you may not have a really good record of the analysis steps you went through. And so it may not be as reproducible as something like writing code in R, or even doing an analysis in Stata where it's saving a log in the background of all your analysis steps and choices. Now, you guys are sitting in the audience, and I know you are a highly quantitative bunch in general. You'll notice those don't add to 100%. And that's because in 38% of articles, nobody was telling us anything about what statistical programs they were using, which is a little bit alarming. And I think against many journal practices at this point. Yes, Ron.
So again, these are proportions of the redacted articles.
Yes.
So, you know, if in a particular field, 20% of the published research is using SAS, but only 4.8% of redacted articles are, that's actually really good, right? Because it suggests that so it's sort of not having that reference to how frequently these are used overall makes it difficult to know if they're protecting against redaction or if they're, you know, more likely to be related to redaction. Is that
true? Yeah. So to repeat for the people online, we don't know what these proportions look like in literature that's not been retracted. And so maybe, you know, if SAS is 5% of retractions, but it's 20% of unredacted or unretracted articles, that might be good. So, right, we don't quite have the comparison group we would like here. I also think that if we were to then say to all the journals, you have to put in the statistical software you're using, it's then informative, but it's not actually indicative of maybe whether or not people are thinking about whether that's an important thing or not too. So I'm not sure it's really causal by nature here in any way. It is more indicative of the attention paid to some of that statistical software and choice. Okay, so who is involved in the retraction? Well, this is pretty interesting. Authors over half the time are involved, and editors are right behind that at 43%. And one of the things that is interesting about this one is editorial involvement was about half in this case of what it has been in similar reviews, but where they just looked at articles that were retracted for things like unethical behavior, falsification, manipulation of data. So editors are involved less than maybe in some of these other egregious situations. Authors were pretty involved. And in fact, it's most often the authors that came forward and said they discovered the error and contacted the journal. This is also a little hard to tell, though, from the retraction reasons themselves. This is now a review of the retraction reasons. Because in 30% of retractions, it's actually not stated who came forward or who identified the error. In 11%, it was an unnamed entity. So there was clearly someone or something being referred to in the retraction notice, but it was never identified as an author or an editor. And then readers were 6% of the time, which is pretty interesting in that, you know, people are reading it and maybe not as frequently as we would like, they're writing in about it or they're able to discover the error. This is mostly sort of author identified. And were the authors involved? So some of the retraction notices state whether or not the authors were involved, and in the vast majority of cases, the authors were. And none of the authors said in the retraction statement, at least, that they disagreed with the retraction. Some of the statements had cases where the authors, there had sort of been a lot of discussion and a disagreement about whether or not the article should be retracted. And that was about 8%. And then there was another 11% not stated. Okay, so now this is now where we're really getting to the part of things where we're like, now we can finally get to the data we're interested in. What were the errors that happened, right? This was a lot of characteristics of things. What were the things that went wrong? So we went through and did a qualitative coding of all the retraction notices to try to understand where they were talking about those errors occurring. And so, and we can kind of split up these qualitative codes into sort of 3 stages of the research process where we can think about getting data, analyzing data, and presenting the results of our data analysis. So in the getting data category, we had 3 sort of themes that we identified and we coded retractions by. One is there were problems with data collection, capture, or the data was just unreliable in some way. The second was data lost or not available. And the third was something with the study conduct or protocol violation. And I will note here, data lost or not available. Now, I think that can be data lost, like, oh my gosh, the data is lost. A student left, the postdoc accidentally deleted the files, investigator accidentally deleted the files. No harping on postdocs. So, you know, but part of me is like, the data is lost. Like, actually, we faked it and we don't want to fess up to it. So we're losing the data because that is a better thing to say. So some of these are not really clear about the intent or all the sort of reasoning behind what's happening. Okay. Analysis of data. This is where I was really interested in the reasons, right? What happens when we're cleaning data, prepping variables, or what can happen in our application of statistical methods? And then we've got the presentation of results. Well, there's the selection of reporting of results. There's concerns about images or figures, which is another one of those places where you're like, is that doctoring the figure, or is that really just sort of a general concern that, whoops, we had the wrong figure in there? And then there's this general catch-all of the results could not be reproduced, which I'm not really sure where it lands, but we put it there. So you can think about where you're expecting to see most of the errors. This is some of the most common ones. So in that getting data category, we had about half of retractions qualified there. And I will say this was not mutually exclusive. So some retractions, you know, there were multiple problems, they got coded multiple times. So none of these are mutually exclusive. Analyzing data, about a quarter of the retraction notices hinted at problems with analysis or data coding, data prep. And 42% had problems in the presentation. And then some of the most common ones are bubbled there. To show, you can see that concerns about images or figures 23% of the time, that was something that came up. Also, data collection, capture, data unreliable, 30%. So now what I'd like to do is go through just a couple of examples of each of these from actual retractions so you can see what it was, because this is what we were really interested in getting at. All right, so data collection and capture. I'm going to kind of go through the get data, analyze data, present data format. So we discovered a technical error in the measurement of this thing. Well, that's legit. I can't necessarily do anything about that from my end in terms of designing software for data capture and analysis, but it's really good that people came forward. And there are some cases where people say, well, we went back to the lab that generated this, there was an error, there was a calibration problem, whatever. So those are kind of errors incoming that it's good we notice. I'm not sure I can do as much about. Here's one where I'm like, I feel like I could help prevent this one. There were 60 children in the study. The ages were by accident duplicated between the upper and lower halves of the database. And you're like, what? And it was like in the same order too. Now, I'm really curious as to know how that happened. One of the reasons we put this in kind of the data collection, data capture, is it would be very hard to have this happen if you were using a data collection tool like REDCap. It would be very easy to have this happen if you were storing your data in Excel and you had a few keystrokes went wrong, or you were looking at missing data and you accidentally copied in or something. So I feel like good data collection practices could have prevented this problem. Here's the data lost. The raw data from 2 papers, this was a meta-analysis actually that got retracted because 2 of the papers it included had big problems, are no longer available, having been lost as a result of a computer failure. Tragic, if it's really true. Certainly a reminder, put your data someplace where it gets backed up. You're not the only person with access, things like that. Good data practices. All right, here's some of the ones I really get excited about. Data cleaning and variable preparation. Here's one that's from JAMA. The errors were due to misclassification of the 2 intervention periods. That is a super simple error and probably a pretty easy one to catch. It was caught later. And one of the things that I really like about JAMA, and one of the reasons I show this, is they actually have, for some of their articles that are retracted, yellow highlights on the retracted version showing what was affected by the wrong data, which is really great information. And they tend to have some of these really nice, helpful descriptions of what happened to say, look, this was a pretty simple error. This was the problem. Here's another one that was pretty simple. There are no concerns about the data themselves, but oops, we switched the experimental and control groups. And that sounds like a simple one, unless you're running a placebo-controlled, double-blind, randomized study, and only certain people know who's group A and who is group B. And some of that information gets crisscrossed along the way. And there was a pretty well-known example out of Hopkins a couple of years ago where that happened. They wrote an editorial about it, and then they wrote about some of their guidelines to try to make sure that doesn't happen again when they're doing their statistical analyses. Now, so now we'll get into application of statistical methods. So here's one. It was a meta-analysis. They use standard errors instead of standard deviations. I have to say that in statistics, sometimes we really hurt ourselves by naming things in a bad way. This is one of those examples. We have standard errors and standard deviations, and the standard error is actually a certain type of standard deviation of a sampling distribution. And so sometimes people see them and think they're the same thing, and they're not, especially if you're doing a meta-analysis. Here's another one, and this is by far the most complex one we found evidence of. This was a case where they had used something called a robust, hold on, I want to make sure I say, yeah, robust variance estimators in this case, which is supposed to be a better way to estimate your variance if you don't know certain things about your data structure. But it turns out it sort of biased some errors in a particular way because of the data in this set, and the wrong conclusion was reached. This was a really interesting one in that it was actually discovered by looking at a figure and saying, I just don't think those error bars can be right. And then somebody wrote to the journal and the author said, no, they're right. The statistical reviewer said, no, they're right. And then somebody else came in and said, actually, this is a case where the robust variance estimator doesn't make sense. Now, this was by far the most complicated thing out there. It is amazing to me that this got discovered, right? Because most of the time people are like, yeah, I've used a robust variance estimator. It's not like any red flags go off. In fact, that's sort of a good sign. But that was kind of like the sort of basic and the most complicated statistical error we found. This was really kind of the most common one, which is a little disappointing, I have to say. All this work, and there are a lot of articles that say there was a major problem with the analysis in our study. I don't know what to do with that. I wish I had more information. Did you use a t-test when you could have used a chi-squared? What was the problem? What were some of the more specifics here? But we just don't get to know. And then there's some of these last categories about selection and reporting of results. It sounds like that could be super biased, but this is where a case or like sort of gets at some of this malfeasance or something. But this was a case where I thought it's a pretty easy mistake to make. The authors had an intent to treat analysis that they had done, as well as a per protocol analysis for a clinical trial. They reported the wrong one. So that's certainly a thing that can happen, especially if you have, let's say, data that's from, you know, an analysis with your missing cases or your complete case analysis, data with some, or an analysis with imputed data, you have them both sitting there. Maybe you look at the wrong one. Maybe you haven't labeled them perfectly. So if you have multiple versions, and they may not differ very much either, except in some ways. But here was a selection reporting problem. Here's the concerns about images or figures. And it was actually really, really astonishing how many retraction notices said there was actually a duplication of figures. If you looked at this panel and this figure and this panel and this figure, it turned out they were the same. Or this is duplicated against this one here, or this is part of this here. So there were all these questions or concerns about images and figures that were really, really surprising, because most of them seemed to me to be things that if you were a reviewer who was familiar with looking at certain types of figures, you would notice they were the same. So I thought this was a little bit of a call to me as a reviewer, to reviewers out there, to say, just check figures, not just to see that they make sense, but look to see if they're the same of any of the other figures in the article. Okay, so just some discussion points, and then I know I've seen some things in the chat. So I'll just remind people that if you do have questions or comments, please send them in the Q&A instead of the chat, and we will be sure to cover them. So some discussion items. So most of these articles were collecting data themselves. So it's really, really important, I think, to think about our data collection process. And errors in the data capture, collection, and unreliable data are the single largest category of root causes that we found in this qualitative analysis. So really important, if you're collecting data, to think hard about making sure you're capturing it right and you're getting the things you think you're measuring. Unfortunately, very few articles report information on methods for data capture or availability of the data. I think if we had more available data out there, we might have a little more, kind of understanding. Well, so a couple things. To make it available, you kind of have to be more careful about how you collect it, even if you're already very careful. And two, then people can see what's going on with your data and sort of hammer at it a little bit more if they think there's something amiss. Data management isn't the most glamorous work, but is really essential to conducting important, robust, reproducible research. And I don't think we always think as much about just the process of capturing that value, writing it down, and keeping track of it someplace. But it's really like one of the most important root things that we do. Now, as we mentioned earlier, there's this new NIH policy on data management and sharing that really may shift practice in regard. I know I've been involved in some efforts to share data, even in like a secure repository, and just the exercise in documenting the data and putting the data together made that data set better for our use as well. Okay. Good news, authors are involved in these types of retractions and may be most likely to identify the problem. In 40% of retractions, authors were the ones who brought it forward. So I think it's really interesting here that the cost benefit to science For science, it's so much better if people point out these errors. But of course, that comes at some personal cost to the scientists themselves. And so I think it's something we ought to consider when we think about, things like pressure to publish or promotion or things like that, that if people have found an error, they shouldn't necessarily be punished for coming forward and saying, that thing I told everybody about, turns out it's wrong. That's part of the scientific process. What we do is hard and complex. And if we expect errors to never sneak through, I think we're being naive. The important thing is to find them and correct them and be transparent, hopefully, about how they happen. Errors in data capture and data preparation are way upstream errors. By the time they get to me and thinking about data analysis, they have happened. And as my colleague Jody Cellino likes to say, you can fix bad data analysis. You cannot fix bad data. Okay, reviewers and editors. Gosh, really, we need to pay attention to those images and figures, thinking about what else we want to know about data capture and data collection. And I think there's some importance here too to saying, statistical review for articles seems like a pretty good idea, that there may be at least some attention to pay to them, some of those things. Excuse me. One thing I want to say about statistical errors, I was hoping to get more information out of this about, 0-1 miscodes or misclassification, or, we used a logit when we should have used something else, or some of these things that I spend time worried about, like unstructured correlation matrices. And we didn't see a lot of those. And I think some of that upon reflection is really that some of those errors may be harder to discover without available data. I also think that, we don't want to go chipping away at good method choices people are making. Instead, what we want to do is correct the really egregious cases, right? We don't want to use t-tests where we should be using chi-squared. We don't want to use simple linear regression when we have repeated measurements on subjects. So those are the types of errors I'd like to prevent. Okay, limitations. I think this is particularly important here because, as we noted before, there are definitely some limitations to what we've done. First is retracted articles encompass only a subset of articles with errors. Certainly, there are many more articles out there that have errors in them. Whether or not those are really important or serious errors, I can't comment. But retractions often come from some notice of obvious errors or something else that happened that made somebody else suspect it. So they're a pretty biased sample of errors. It's also the case that high-profile articles may just get more scrutiny. So maybe, with a higher profile article, you're held to a higher standard of publication, but people may be looking at it a lot more. We don't have a control group of articles yet. So one of the things I'd like to know is, does the statistical software used in a matched set of articles differ if they're not retracted? So that's kind of something we have slotted for some future work when we recover from all our full article text review. And in fact, I think what we'll actually do is probably pick the articles like with journal impact factors above a certain level to really get into that a little more. Retraction notices. I was at a conference a couple weeks ago where Ivan Aransky, who's the one who really established the Retraction Watch database and works at the Center for Open Science and with the Center for Open Science, we were talking about retraction notices, and they are super political and even rather legal in terms of what they can say. And certainly the absence of transparency in those retractions inhibits our ability to learn from the problem. Passive voice and euphemistic language are commonly used in the retractions themselves. So sometimes you read it and you just can't tell what's going on, even though there's something being said. All right, so with that, just a thank you to the team here. Because of the whole time during which we conducted most of this, it was very hard to get all of us together for a picture. Oriana isn't pictured. Grace is kind of badly photoshopped in there. But this work is really a credit to these folks. There was a ton of review, a ton of effort. There's some of the software we've worked on developing. StatTag is out there. StatRep, like I said, is in development. I'm happy to talk to people about being testers or users as we work that out. But with that, I really thank you for the chance to be here and welcome your questions. I know there were a few little bubbles up in the chat, so maybe there's some questions in the Q&A we could start with.
Yeah, so we have a question from Melissa Gesbeck. It's just a comment on the capabilities of SPSS. A log is automatically generated during point and click analysis. You may also code it manually using a syntax file.
Great. So that's wonderful. I am glad to hear that. Thank you, Melissa. So certainly stated as a log, it sounds like SPSS does as log too, which is fantastic to hear. I hope people are saving their logs. Questions from the room? Yes.
Did you look at the impact factor of these 700 plus articles that were redacted?
We have not. The question was, did we look at the impact factor of those articles that were redacted? We have not looked at the impact factor in particular. Having looked at a lot of those articles, I can tell you there's journals you would recognize and you would be super happy to have a paper in. There are also journals I was like, wow, I didn't know this existed. And it seems like a very sort of small niche-y place. So we really were across the gamut. I have some suspicions too about some of the most frequent flyer journals there, but I wouldn't want to comment. But yeah, so I think we may look at that. Questions online?
From Maria Pyra, do you think that there's a role for double coding and or publishing analytic code?
Absolutely. Is there a role for double coding? Yes, I think that's really important. In fact, we double coded our abstract review in this case because we wanted to make sure we all agreed that we were getting abstracts that related to clinical and translational science. That's pretty easy to tell if you have human subjects research. It's much harder to tell if you're reading a basic science article and you don't know a lot about basic science and you're trying to figure out if it really relates to human medicine or human health, or if it's really very much in the basic science realm of things. So we double coded here. I think it's really important if people are really interested in a particular outcome or particular thing, they should try to double code. I think when that's not possible, and obviously it isn't, it's really important to have things like REDCap where you can have logic checks on whether or not your values are reasonable. And the second question was publishing analytic code. Yes, I think it's a really good idea. I think it's something we're going to start to learn how to navigate better and better as we come across this NIH data sharing and management policy that, you know, it'd be nice to share your code and show other people what you've done. And the code is a very clear indication of how the analysis was conducted. You have to be very careful that you don't have participant identifier information in the code itself, which sometimes we do, you know, to say, excluding these 3 cases because they don't qualify, blah, 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 or something, and you actually end up with things that are identifiable in there. I really like the idea of publishing code. One of the things we do in the Biostat Collaboration Center, because we work on so many different projects and different people touch them sometimes, that we try to make it so that our code is such that somebody else can come in and read your code and know what you did. So it also needs to be heavily commented and structured in a way that people can follow it. A question from the room? Yes.
So like where can we go to if we're, let's say, like making an experiment or analysis and the statistical analysis has already been selected? No, for example, like I don't have robust or like well understanding in statistics, so like is there like a second opinion I can get? Like someone else like
Yep. So the question was, let's suppose you're here at Northwestern doing an analysis and you think ANOVA is the right thing to do, but you're not quite sure and you want a second opinion. I run this thing called the Biostatistics Collaboration Center. It is a collection of 12 faculty and I think 8 or 9 master's level biostatistics staff who have a lot of knowledge about whether or not certain decisions are appropriate. And we have an online portal where you can fill it out. And everyone gets an hour initial consultation to discuss things. Usually those are discussed with the idea that, you know, maybe we're going to go on and write a grant together and we'll write the grant together, or maybe somebody has an analysis they want us to conduct and we conduct it. But people can use that time to come in and just sort of do some brain picking on, well, I think it's ANOVA. Does that seem quite right? I will say there are some limits to that sometimes there are weird features in data that you don't know until you actually open it in a program and start to look at it. So there are limits to that advice, but it's certainly available. I will also address the fact that we've been super, super backed up in terms of those requests, but we've just hired a number of wonderful, fabulous new analysts over the summer. And so they're joining us and we're steadily chipping away. So it's not an instant answer, kind of give me an answer right now, but if you have some time and a runway, it's certainly something where we could try to help out. Any online questions? Oh, okay. Any other questions from the room? All right. Well, thanks very much.